next speaker is from University of Southern California, Cong Zang.
Hi everyone, my name is Cong Zang, and I'm a third-year PhD student in University of Southern California, Los Angeles. And it's my great honor to be here to introduce my study entitled Brain Age Gaps Derived from T2-FLAIR MRI Wet Matter Capture the Unhealthy Aging Related to the Cardiovascular Risks. It's freezing. Sorry. It's not a touch screen. Sorry. Okay. Okay. Thank you. Sorry about that. Yeah. So starting from the introduction, the brain age is an aging biomarker measured at the neuroimaging level. By training the machine learning models using the training data set, which should be the healthy subjects, we are able to predict the brain age on the testing data set, which could be either healthy or unhealthy subjects. And we are able to calculate the brain age gap, which is the difference between the predicted brain age and the chronological age. So the brain age gap is very important because it reflects the level of neurodegeneration. From this figure, we can see that for the blue dot, the brain age is higher than the real age. So it indicates a positive brain age gap and a potentially accelerated aging. Since there are so many structures in the brain and all of them may undergo different aging trajectories, especially for the gray matter and wet matter, which are closely interconnected but may share different aspects of aging throughout the adulthood. So some of the aging factors affect both gray matter and wet matter, such as smoking and alcohol. While some other factors like Alzheimer's disease affect mainly the gray matter, while the CSVD, the cerebral small vessel disease, affect mainly the wet matter. So to better understand the gray matter and wet matter aging separately, in OHBM 2023, we've had an abstract showing how brain age gaps derived from T1 and FLAIR images capture the different aspects of brain aging. We found that the T2-FLAIR derived brain age was associated with cardiovascular risks, but our 3D CNN model did not separately analyze the gray matter and wet matter aging due to its black box feature nature. Therefore, whether this finding is driven by solely the wet matter changes or both the gray matter and wet matter changes remains unclear. So here we hypothesized that brain age gaps derived from T2-FLAIR signals sampled at various depth levels within the wet matter represent the wet matter specific brain aging. To achieve this, we are using a graph-based analysis. It should be noted that our graph-based analysis ensures fixed topology, points correspondence, and very good interpretability. Since this is a pretty complex figure, I will break down and explain every part in detail. So the first part is actually step A and B are the data preparation and pre-processing. We are using the UK Biobank data here, the 3T MRI images, both T1 and T2-FLAIR. The UK Biobank data is world's largest human studies with all kinds of measurements, including MRIs. So we firstly did a wet matter surface reconstruction using the CVAT pipeline from MNI Canada. And we did a whole brain segmentation using the SynthSeg, which is a deep learning-based whole brain segmentation method, now publicly available on the GitHub or on the new versions of FreeSurfer. We also did a linear registration from the T2-FLAIR to the T1 image of the same subject using the FSL. So next, we developed a medial surface reconstruction pipeline by shrinking the original wet mat surface all the way down to the ventricle surface and generate 9 medial surfaces along the path. So firstly, we generate the Laplacian field, which could be regarded as a distance map, which means that the vertices on the wet matter surface that are far away from the ventricles, ventricle surface, will be moved much faster during the deformation, while the vertices that are closer to the ventricle surface will be deformed slower. So this ensures that all the vertices reach the ventricle surface in the same speed, and it preserves the topology of the mesh, which is also called isomorphism, which is a very important property during the surface reconstruction. We then did the actual surface deformation by controlling the depth ratio, which measures the spatial locations of the medial surfaces. So 0 means that it's exactly on the white matter surface, while 1 means it's exactly on the ventricle surface. And in total, we generated 9 medial surfaces ranging from 0.1 to 0.9. And we used those medial surfaces to do the feature generation by mapping the white matter, the medial surfaces onto the T2-FLAIR images and subsample the white matter intensity and form the feature matrix. So the size of the feature matrix is actually 5,000 by 9, which is the number of vertices by the number of medial surfaces. And then we did a brain age prediction using the 5-graph convolutional layers and 2 fully connected layers. And again, the brain age gap is calculated by the brain age minus the chronological age. So finally, the statistical analysis. We are doing the multivariate GLM by correlating the brain age gap with confounders and main variables, including the habitual, cognitive, cardiovascular, and neurodegenerative variables. For the training, we have 9,000 healthy subjects, and for the testing, we have 800 healthy subjects and 2,200 subjects with all kinds of risks and diseases. So here are our primary results. First, our model shows pretty good performance with a mean square error of 3.08 years on the validation dataset. And the GLM results. So the x-axis refers to the beta coefficients, and the stronger the higher the beta coefficients are, the stronger the effect the factor is to the increase or decrease of the brain age gap. And we can see that, especially for the cardiovascular risk factors, the increased heart rate, increased systolic and diastolic blood pressure, history of hypertension, increased white matter hyperintensity volume, and increased BMI are significantly correlated with increased brain age gap. And by the way, the red square refers to the significantly correlation. So to summarize our method, our pipeline was designed to effectively estimate aging within the white matter by sampling the T2-FLAIR signals at various steps. We proved that white matter specific brain aging modeled using our pipeline significantly correlate with various cardiovascular risk factors, suggesting the importance of separate white matter brain age models to investigate the pathway involved in cerebral vascular disease. Certainly, there are some limitations, like the brain age prediction in different structural regions. For example, the periventricular white matter versus the deep white matter, and the functional regions, like frontal versus parietal, has not been investigated. So I would like to say thank you to all my precious lab mates and collaborators. And if you are interested in our study, feel free to email me. And that's it. Thank you for listening.
We have a few questions. The first question is, do you account for any covariance when calculating brain age prediction? Age, race, for example, race, sex, scanner types, scanner site?
Yeah, that's a very good question. So we've got all those confounders shown here, like the scan site, the sex, and ICV, intracranial volume.
And the second question is, by constructing, the model depends on the enlargement of the ventricle. Could we then call this approach a white matter brain age?
That's a very good question. So actually, we are not using the enlargement of the ventricle itself. So we are basically doing the brain distribution based on the white matter. And by the way, the brain distribution model using the GCN does not take into account the size and shape of the brain, including the ventricles.
One more question. Do you correct the bag against age distribution bias?
Age distribution balance?
Age distribution bias.
Age distribution. Yeah, so we firstly, I think if I understand it correctly, you're asking about the difference between the training set and testing set. So I did a correlation test on the training set and testing set, and I didn't see any age bias in those two data sets. Does that answer your question? Yes?
Let's have one more question. Sorry, I see that's first come, first serve. Sorry.
Maybe I didn't see it clearly, but you had a figure that showed there is no significant brain gap for dementia. Do you find that surprising? Do you know why that would be?
Yeah, that's a very good question. So somehow, like, dementia has been shown to be related to white matter in these years, but we still regard it as a primary gray matter induced disease. And of course, this is a multivariate study. So if suppose we are doing univariate, it could pop up some significance, but that's worth it to be investigated. Thank you.
Thank you. Let's thank to the speaker.
Thank you.